So with the transduction, I said that there's two methods. You've only learned one so far, but that's the generalized one. So can you think why do they call it generalized? What are they zooming in on here that where they're saying, oh, it's generalized? Random, very good. Random. It's all random. It's like a crapshoot. What stretch of DNA is gonna get packaged? Because as that virus is taking this cell hostage, one of the things that is that you can get the breakup of the chromosome like that, the host chromosome. And it's just, they're all just, you have to imagine, all of those bits and pieces are floating around and it's totally random. As the virus is putting itself together, it's totally random. Totally general, which structured DNA will get put in there. Very good. So that I'm making a big deal about it because the next version we're going to see is not generalized, it's specialized. So with transduction ideas, you have a third player in the game. You have a first host and you have a second host and you have a virus. That's the middle person that can do the transfer. So remember now, the first host is toast, right? It's not going to make it. So here, that's what you're seeing where you have the virus taking that cell hostage, make more of viral particles, make more of me, make more of me. And so you see all the particles getting made. And then here you have the assembly stage where all the viral particles are assembled into coherent viral particles. And then after that, the first host lyses, breaks open and dies. So we're not really too focused on that, but we are focused on the idea that some of these viral particles contain the first host DNA. And then when that breaks free and finds a nearby cell to infect, it's defective. And what happens is not only is it transferring some of the first host DNA, which could be really good for the second host. And it's not really technically a host like gas, because really what happens is it'll transfer that DNA, the DNA gets incorporated into the chromosome, and usually there's not enough viral DNA in there to do any damage. And so this second bacteria that gets attacked by a virus, it's like a abortive attack. It won't be able to, the virus won't be able to finish its goal. But this second bacteria now might have some useful DNA from the first bacteria. Does that make sense? Okay, so we have this, could be a big payoff for this second bacteria here. Okay, so that's generalized, totally random bits of DNA floating around, and it's random which ones will get packaged into the viral particle. Let's go at specialized transduction. Now, specialized transduction, there's going to be a special piece of DNA that will get transferred. It's not random anymore. And here they're showing a bacteria, and they're not showing the virus attaching and injecting its DNA. It's already done that. And the reason I know that is that they have it labeled here, phage DNA. Okay, so when someone labels a cell like that, when someone labels a cell like that, I'm trying to see, no, stop. Erase. All right. There we go. When someone labels a cell like that, if it says phage DNA, another word for a virus that infects bacteria are bacterial phages. Some people say bacteriophages, but I don't. I say phage. Each their own. Bacteriophage, and then we just short it to phage. So that's, if someone says it's a phage, it's a virus that only infects bacteria. It won't infect animal cells. All right, and it says phage DNA. So what has happened already, the artist didn't show this. The virus injected its DNA, and the DNA got incorporated into the bacterial DNA. Got pasted in there. And then if that happens, I would say, oh, look at that bacteria. It's been lysogenized. Right, so that's how you would talk about it. So a lysogenized cell, if I say that, I know that cell has phage DNA, viral DNA, pasted into the host DNA. And then we say the bacteria is a lysogen. You can say it that way too. You can say, oh, it's a lysogen. And if you say it, then what you're trying to get across in one word is that you know that there's phage DNA pasted into the host. Okay. So why do they call it specialized transduction? Well, compare the two here. So the phage DNA is green, and then the host cell is, it's like lime green, and then pink. So in a normal event, what you can do, what you see is sometimes the viral DNA that pasted in there like that, and then it just stays in the DNA for a while until some event happens to cause it to get cut out from the DNA. So that's what you're seeing here. This viral DNA, the phage DNA, is getting cut out. What color is the DNA here? Green. It's all green. What color is the DNA here? It's not all green. All right, so this is specialized. They're saying every once in a while, it's not hugely common, but every once in a while, when that cell, when the phage DNA becomes active and it starts to cut out of the chromosome, it's sloppy and doesn't just grab just the phage DNA. It grabs special DNA, which is the flanking DNA, the flanking host DNA. Not just any piece of the host DNA, but the flanking piece, the piece right next to where the phage DNA is. So it's special DNA. Right, so that's the difference between the general and specialized that they're trying to get across here, is that you have the DNA that's flanking the phage DNA that's going to get cut out kind of accidentally with it and brought as part of the phage DNA. Now you have to imagine if that phage DNA gets cut out and it has a little bit of flanking host DNA. What happens is as soon as the phage DNA, either way, when the phage DNA gets cut out, then the active cycle of viral replication starts. And so now the virus takes that cell hostage, says make more of me, make more of me. And that's what you're seeing here, more viral particles. Except now some of the viral particles here, they have not only their own original phage DNA, but some of the host bacterial DNA too. And if this virus breaks out of that first host and gets to a second bacteria, it can inject that DNA, and here they have it labeled defective, okay? Defective phage. What they mean is there's not enough viral DNA to kill that second host. It's just kind of bringing, it'll be able to bring that DNA from the first host with it and inject another nearby bacteria, and that can get pasted into the bacterial chromosome. And now that second bacteria has DNA from the first bacteria, first host. And so the virus can't really take that second host hostage. It doesn't have enough of its own genes to do that. And well, I should say it's defective. It's the more proper way to say it's defective. And so essentially in either case, whether it's general or specialized, that second bacteria gets access to bacterial DNA from someone nearby. And if everything goes okay for the bacterial perspective, it won't get successfully infected by the virus and killed. So therefore it's going to, hopefully take advantage of that DNA. And if it helps the bacteria survive, great. It'll maintain that stretch of DNA. Does that make sense? Okay. Yes.
I just had one question. So the very beginning of specialized transduction, would that be the randomized part? like if it goes normal or rare, but then
Mm,
it's non-randomized because it like duplicates itself. You know what I'm trying to say?
So with specialized transduction, I guess. You can think it's special in two ways. It's rare, so that's special. Because it's rare for an imperfect cut out of the DNA. And then it's specialized also because flanking, very special pieces of DNA will get packaged. It'll be the flanking DNA on either side of the phage DNA. I'm not sure I answered your question correctly,
Oh,
to what you wanted.
yeah, it's all right.
Other questions?
this image here is putting generalized and specialized on one comparison
Figure for you to look at as you're studying. Oh, I forgot to introduce that word too.
Is it? Let me go back here and I'll just look and see if it's here. So it's not there. Interesting. All right, so it's only on this slide. All right, I want to point out another word here. Prophage. So this is a term that they give specifically, so I said phage DNA, phage is referring to viral DNA. And then if they use the word prophage, they're being very specific in saying it's definitely pasted in there. Okay? And it's securely in the host DNA, then they'll say it's a prophage. So there's the reason they gave it a special term is because we know that there's some bacteria, well, there's a lot of bacteria actually, that have viral DNA like pretty permanently pasted into the host chromosome. It's just part of it. And so then you call it prophage DNA. So they've mapped out and sequenced the DNA. They know that's a viral stretch of DNA. And actually there's a lot of bacteria out in real life that have that. Just like, believe it or not, we have a lot of viral DNA in our chromosomes. But instead of calling it prophage DNA, we call it provirus DNA. And that means it's in our chromosome. So be comfortable with that word too, the prophage idea. But this is a side-by-side comparison with some extra text as you're studying. Okay, so this is the most complicated, primarily because there's another player in this game. It's the virus that is kind of playing the middle role of transferring the viral, the host DNA from one bacteria to the next. So, bacteria are not totally at the mercy of viruses though. And under natural conditions, they have some ability to defend themselves. So they do have their own version of an immune system. Certainly not like our immune system, but you can imagine if you are a bacteria and viral, a phage latches on to your surface and then starts to inject DNA, you might not think that's such a good thing. It could be a really bad thing for you. And so having a way to try to stop that infection process. And so they do have that ability. And you may have already kind of peripherally done some work in like intro to cell and molecular. If you worked with restriction enzymes on any of your protocols, restriction enzymes, you can thank bacteria for that. Bacteria have a lot of restriction enzymes that have been isolated by scientists and stored in the freezer, and then you go and buy them and they ship them to you. And the restriction enzymes, they often use a picture, I think mine has a picture of scissors, because it will restrict pieces of alien DNA that's coming in. So as that viral DNA is coming into the bacteria, if the bacteria is up and running and knows what's good for itself, it will most likely try to cut up those pieces of DNA, because if you can cut up the DNA, it can't function. It won't allow for the virus to take the cell hostage and make more of itself. So we call them, the fancy word is restriction endonucleases. I tend to say restriction enzymes, just a little shorter. So you can buy those, like I said, for intro to cell and molecular. You probably may have used those in some of your experiments. Another thing that bacteria can do is they can try to modify DNA. Whoops. So here they can add different things onto their DNA, like a methyl group, CH3. So methylation. We do that too. Our chromosomes can be methylated to protect stretches of our DNA. And so here they're trying to show, here's the restriction enzyme idea with the scissors. And they're showing this virus trying to get that cell hostage, and they're cutting up all that phage DNA will make it useless. And then here they're using the yellow balls to show methyl groups. And again, if you have protected sites like that, then it's easier it's like a basic immune system for the bacteria in this regard. And so it's a way to protect that stretch of DNA from getting accessed essentially and expressed or cut into. So when they saw that there's viral DNA embedded in bacterial DNA, they started looking to see if that's, was it just happening under lab situations when they were experimenting or not? And what they discovered was rather surprising. When they isolated bacteria from all kinds of different habitats, they found that there was phage DNA. And so they were prophage, and then you'd say those cells are lysogens or they're lysogenized cells. And so the idea here is, if so many different cells have this, then maybe there's some advantage to having phage DNA. So this is a little, here you're seeing the virus appear, attaching and injecting its green DNA, and you can see that with the lysogenic pathway, the viral DNA goes and pastes into the host. So that's your prophage idea. So if that stretch of DNA gives an advantage to the bacteria in some way, and I'll explain what some of those options could be. So sometimes viral DNA, just viral DNA, can be useful. In this case, to the bacteria. It can be useful to humans too. Some of the viral DNA that's in our chromosomes, for example, allow us to have a mature placenta to support a developing fetus, strangely enough. So sometimes this viral DNA in and of itself could be useful. And so if a lot of different bacteria that they've isolated have prophage that's stable, that's the hypothesis. Maybe that stretch of DNA is useful for the bacteria. The other kind of cool thing here is once that viral DNA inserts into the host and becomes a prophage, and then that cell goes through cell division, it'll just transfer, it makes a faithful copy of the whole chromosome. And both offspring get that. So now the offspring have that viral DNA, the phage DNA. And so you can pass that on to subsequent generations. And from the viral perspective, that's kind of slick too, because you don't really have to do anything. You just become a prophage and you're infecting progeny over and over and over as they go through the generations. And then eventually you might activate. And so it's a way for it's kind of a dual double-sided knife idea where from a prophage perspective, they're spreading into future populations. That's good for the prophage because if it becomes active, it's got a lot more cells that's infected in a very passive way. They didn't have to build this funky structure and keep, infecting and go through all that. They're just there. And they just get copied and put into the progeny cells. So it's kind of slick from the phage's perspective. On the other hand, if that stretch of DNA has some use to the bacteria, that's good too. And so it really is context driven, like what is that stretch of DNA and is it really useful or not? So here you can see that something happened where this prophage cut out and became active. So they refer to it as the induction phase. You've induced that cell to have an active viral particle, and then that cell starts to, it's taken hostage and made, just make more viral particles, and then usually dies once the virus is mature and breaks out. You can do that in the lab. The way they usually induce cells is they hit it with like ultraviolet light that usually stresses the cell enough where the viral, I like to think, I don't know how deeply they've looked into this, but to me, it seems like the virus is like, I'm jumping ship. The cell is stressed out and I'm getting out of here, And so if you stress the bacterial cell in some way, typically it's some kind of toxic chemical, UV light, or you starve the bacteria, then you can get the prophage to cut out and get active. So they jump ship and try to find some other host. Okay, so let me give you an example. There are some bacteria out there where once the phage DNA inserts into the host DNA, there's stretches of genes on that phage DNA that allows them to produce toxins, where if they didn't have that phage DNA, they couldn't produce the toxin. So there's a classic example, Corynebacterium diphtheriae, which causes diphtheria. If you just isolate a normal Corynebacterium diphtheriae out in nature, it might not have the prophage in it. And if you got exposed to it, it'd be totally fine. You wouldn't get diphtheria. And so they know that it's the pathogenic ones are the ones that are lysogens for this particular gene. It's called an exotoxin gene. I'll talk to you more about exotoxins later. It's nasty. Okay, it allows for the Corynebacterium to become pathogenic. And so there are certain species out there that don't have that viral DNA, so they're fine. But then there's others that are considered pathogenic. Another good example is Clostridium botulinum. That one causes botulism, very, very deadly. And the phage DNA has a gene on it that codes for a neurotoxin. And so again, some species of Clostridium botulinum are totally fine. You could be totally exposed to it and it wouldn't matter. But if you have the exposure to the one that has the phage DNA for this gene, and I have down in the notes this neurotoxin, as little as 0.000005 micrograms. That's super dilute. Okay, that's 5 zeros plus the number 5 micrograms. Can kill a mouse. 1 ounce could kill the entire US population. That's how toxic it is. It's the most toxic biologically made substance we know of. And yet people inject their faces with it. Botox. You better hope the person administering that did their dilution correctly. Can you imagine? I sure hope they have computers pumping out the dilution scheme for this. You did serial dilution, right? Yesterday. Can you imagine if you got your pipette skills a little off? Yeah. So, but yeah, Botox, that's
So when you get injections of Botox, apparently what it does is it paralyzes that tissue. And that's the neurotoxin. It paralyzes. The reason you die from it, if you get exposed inappropriately, is it causes your diaphragm to become paralyzed. And then you can't suck in air. And so essentially you asphyxiate because your muscles go into spasm and paralyze. And on that happy thought, but on the other hand, it's been put to, better uses. Apparently it reduces lines on your face. Well, yeah, you're paralyzed. You can't like express. I don't know. That's gotta be really weird. Like you want to laugh, but you can't because you're paralyzed. I mean, that's seriously, you can't smile. You can't do an, surprise where your forehead creases. You can't do that either. I don't know if you ever heard of the show, it's a comedy show. They do reruns. Frasier. They did a whole episode where Frasier's brother, Niles, got Botox and then they went to a dinner party and just I was laughing so hard through that because he was trying to express himself and he couldn't because he's all paralyzed. So anyways, so there's a downside to Botox injections, but on a less silly, actually quite a good use of Botox. They have used it for like Parkinson's patients to settle the tremors. And so there are very good uses for that. But again, super, super potent. All right, so it just, so in these cases, maybe, being pathogenic for Corynebacterium and Clostridium is a definite advantage for them. It could give them an advantage where they could get better access to food. Unfortunately, it's usually a human that they're feeding off of, but the other idea too, once you've become a lysogenized cell, you're immune to further infection by that same virus. So whatever virus pasted its DNA into you, that same virus, if it came along again, it couldn't infect you. And I don't know the mechanism. Maybe Dr. Sharp would know on that, but you're immune to further infection by that virus. So that could definitely help you because now you won't get multiple infections. And like I said, it could give you some adaptation where some of those genes that come along could provide a selective advantage. Yes.
Like, for example, if somebody's like, oh, I'm immune to this cold that I've already had it.
It's more our memory T cells tend to do that for us. So we have very specialized immune cells called memory T cells, and they're the ones that remember, you've tried to, infect me before, and I've built up a resistance. Basically, if you get exposed to the same pathogen, your body remembers and can nip it in the bud because the cells are up and running and can attack. So now we have a lot of bacteria though that are in our gastrointestinal tract in particular. And so they've started looking at, do bacteria in our intestines, are they doing this kind of thing? Are they doing transformation and conjugation and transduction? Kind of makes you wonder, right? And so if you think about it, there's all these billions, trillions of bacteria that are in your intestinal tract, and they are crowded in there. They are in huge populations where they're not existing in isolation. They're bumping up into neighbors. And so they've been looking at this idea and they can see that there is exchange. Higher frequency, they've done studies. I might get the number wrong. I think it's a 25-fold higher level of gene transfer with bacteria that exist in you than the wild bacteria. So it's like our intestines provides a very good habitat, good conditions for these types of gene transfer mechanisms to play out at a higher frequency. And so they've been looking at that, and primarily one of the concerns, I guess, that they have about this is antibiotic resistance. And so if you are sick with a pathogen that sets up camp in your gastrointestinal tract, can there be transfer of the resistance genes into the general population of your intestinal tract? And so the medical microbiologists are trying to investigate this and see what the situation is. So horizontal gene transfer, could that be going on if a pathogen is able to establish for any length of time in your intestinal tract? And probably all of us in this room have had pathogens that have established in our intestinal tract and has caused us temporary misery. Where, have you ever been so sick, you're in the bathroom and you don't know if you should sit or kneel? Because it's coming out both ends, you know, am I gonna puke or have diarrhea? Which is it, you know? And you just lay on the tile floor and moan. So, yeah, you know, we've all experienced that, I'm guessing. And, you know, as they are existing in our body for that 24, 48 or 48, 72 hours, are they doing any gene exchange? And so they are looking at that. Here's a visual of this idea where they're looking at transformation, conjugation, transduction. So to be continued on that story. They don't know enough data yet on, other than some of the data is showing a much higher frequency, but they're looking more into that. The other thing I just want you to, again, I've said this before, but don't forget that you can have genome reduction going on too. Like I've been talking so far about phage DNA getting pasted in, so you're getting addition or transformation, you know, addition of DNA. Yes, certainly that happens, but you can get loss of DNA too. And so, for example, Mycobacterium leprae is, when you look at its current genome, it's really changed over time as it interacted with the human host. And so it has so-called pseudogenes, which just means they're non-functional now. They've changed enough where they just don't work like they used to. And basically, it's like if you become obligate to your host, where you have such a tight interaction with your host that you don't really live outside your host anymore, then you can get away with getting rid of a lot of your genes and still being okay. A lot depends on how it's spread and all of that, but so don't forget about reduction. Microbes are pretty good at that. They have to be, because if you can imagine large stretches of DNA that are no longer useful, that's a lot of resources and energy to keep that, the upkeep of it. It's like having a huge house versus a tiny house, much harder to clean a big house. That's what I always think when I see these big mansions. Who cleans that thing? I would hate to have to clean that thing. So anyways, gene duplication, again, to remember, microbes are haploid. Their chromosome is just haploid. They have one gene. Gene duplication is a process that they can use to duplicate one of those haploid genes and create situations where maybe that duplicated gene could be used as a backup, or they might mutate that gene. There are mutation events in that gene over time that maybe it's going to allow for something different. So microbes are just very adaptable. They can take in DNA, they can purge out DNA. Mutation events happen at much higher frequency than with animal DNA. And so they have this great flexibility. And that's one of the reasons you should be thankful that so many microbes live with you, because they are helping you because of this very easy adaptability to current situations and current habitats. They help you survive and thrive because of that. We'll talk more about that when we get into that section of the semester. So here's an interesting graphic where they're looking at interdomain transfer of DNA over the course of evolution. So here, the bigger the circle means there's more genes. So DNA sequences from many eukaryotes, including us, appear to include a variety of genes that are horizontally transferred from bacteria. So you have the origin archaea DNA, bacteria, right? And you can see bacteria, Drosophila has 65 genes that come from bacteria, and look all the way to primates, 220 genes. So kind of makes sense when the first ancient life form on the planet was microbial. All right, that's where it all started. And then living cells, protocells formed. It was prokaryotic microbial cells. And then over time you have speciation and diversification. Kind of funky and weird to think about that stretches of our DNA really are from microbes. Kind of cool. All right. Well, that is what I have time for to focus in on gene transfer right now. We're going to come back to the idea of gene transfer in a little while because they've discovered some new methods that actually have a lot to do with how microbes also communicate with each other. So I'm going to save that for the communication. And so the next topic we're going to get into is cell structure. That's the next topic. But before we do that, if you have your phone available, I'd like to try this Google form idea and see if it works. We'll see.